Hello. So when my student put there the title, so this is probably not going to be the title of the paper. And when I saw that it was this big, he was very happy with the name. He didn't want to make it shorter. So I am not the person that should be presenting it here. So I have a brilliant PhD student in a double degree between university in Brazil and university here in Germany. And we got fundings for him to come to Germany. And at the moment, his passport is with the German embassy. So that's why I'm giving the talk. But I always like to give a small shout to the people that are sponsoring and financing the work. We work with a lot with the AI for PEP that it develops, that tries to democratize AI in epidemiology and preparedness for the next pandemic. So this is very important for us at this work. And we are also a use case within the NFTI for microbiota. But so all of that I said, so why should you care about my presentation and how that could connect to you, particularly if you have no interest in proteins or the connection between proteins and microorganisms. So we start looking at one part that is protein language models and using protein language models as a proxy to what we could do with other types of language models, and particularly if we can start asking the questions or think about things that we don't know. And as a person that was not trained initially as a computational scientist, I started as an ecologist. We always try to bring this nature-inspired design to anything we do. So we are trying to do the same with our research in AI. And when we think about what we are going to show is that when you actually have a prototype or something that worked really well in a data set, or even if you use 50, 100 different data sets, when you actually bring this to users, you find out that the things that you planned don't work really as you thought they would. The second thing is that although the people working in ecology, they are used to work with big data for about 3 decades, they usually are interested in the context of one sample within a very large data set. And there is in this picture, there are 2 species of penguins, and I dare you to tell me which are which. So we can't only look at the data and think, okay, can we grasp what it's coming back to us? And many times we go and we think, okay, we are right, but what if you're not? Because let's say when I go and we will publish a paper saying we get almost 200,000 novel proteins, and then you come to a company and say, hey, let's work with it, and like, okay, we can't test all that. And so which ones do you test and why do you test it? And this is something that I'm going to try to talk about this now. So this project is in a context, we have collaborators in 6 different countries, 3 different continents, and a lot of different places, so, but I will not talk about all of them because I would have to put 50 people on the slides. I will only talk about the people in the Helmholtz that are helping. And this project is within another project that is it's an Interpool project called AI MAR Explore. And we have like Brenna and I in the UFZ. Brenna is the PhD student that should be giving the talk. And then we have people in GeoMar that are collaborating. We also have people in Jülich. We also have people in the GFZ. Very recently we were joining with people in Helmholtz Munich, and I always like to give the shout out to my biggest collaborator in the Global South, where we have a lot of double degree PhD students coming up. Another topic, if you want to talk about double degrees and PhDs and how to make this happen, please look for me. Like this is a topic that I'd like to talk a lot. But then we come back to the topic of the presentation, that is, if you are looking for language models and how to use these to characterize novel and to understand and actually what is the function of an uncharacterized enzyme. And so there is a lot of people designing novel proteins. So they go and they're like, okay, we know all about these functions, but what the thing is that we don't know the function of most proteins out there. And if you actually take 1 gram of soil here, we go in the grass outside, it's very likely that you're only going to be able to characterize with the state-of-the-art methods. It doesn't matter what you use. You're not going to characterize more than 20% of all the proteins there. And if you go to the human metagenome, that is like all the microbes that live in your body, we don't know we only know 60% of those genes, so we still have 40% of all those genes that don't have a function. So characterizing those functions, it might be more important than designing a new function, because nature and evolution do a very good job with things, and it's very likely that those will be more efficient than the ones we would design in a computer, at least for now. And when we think about proteins, anything we do is based on alignment. And last night I was talking with a colleague of mine that was saying, hey, I was in the conference with the people that developed the alignment-based annotation, but there is a lot of problems with that, because everything you do is that you align sequences of, and it doesn't matter if it's a nucleotide or a protein sequence, and then from there you have thresholds that right now don't matter, because we are talking about state of the art, so we take the standards from the field, and then eventually you get the function of a gene. But there are several limitations with these methods when you actually give a meaning for it. So let's say that our protein would be our words, okay? That's a lot of things that look very similar. They don't have the same function. And a lot of times you have things that are very different and actually have the same function. And even when you actually are able to annotate, to give a name for a protein, that protein is in a context of interactions with thousands of other proteins, and it may not work in the way you expect in the real world. So how those things are, and of course we saw the key speaker, the first key speaker of the conference. He was already talking about protein language models. We know the Nobel Prize last year from the AlphaFold. And so these ML-based techniques to find functions are in the hype, and I'm not trying to look for the next AlphaFold, but we're trying to understand how we actually think about the data that we use to develop those proteins. And so just these ML-based tools, they go beyond homology, they can go to fine-tuned resolution. We can start doing these experiments to see if substrates can go and be in the right place in the protein. And so there are many advantages over align-based methods. And the question, so when we started doing this 7 years ago, is that can we use ecology and biotechnology to enhance the characterization? So we're not designing new enzymes, we are knowing things that we know we don't know. And so how this is possible? So we should think about foundational models, like not everybody have the power to train things from scratch, and this training takes months and you need GPUs, and most people don't have access to those things. But we should think about the protein language models are to proteins what large language models are for text. And you basically, we hope that the protein language models, we're going to be learning representations for protein sequences, and we are going to not only be able to give an annotation, but also understand the properties of those proteins. And we also can, if we develop a protein language model in one field, for example, cancer research, by the way, proteins are proteins, we can take that and transfer it to microbiology, and that means that if we develop something from microbiology, that could be brought back and moved to anything that actually deal with proteins. So we started not with the protein language models, but most of the work was actually generating data, or at least curating the data that we would be using. And how did we do that? If we would be in an audience that would be microbiologists, would spend like 30, 40 minutes just in the slides and everybody would be happy, but I will go here as fast as possible. So basically we used two foundational models. So we didn't develop the foundational models. So the pros and the protrans. One is BioLSTM, and then basically if you think that you're thinking about past and future, and you actually have transformers. And then we got those and we added the hierarchy, multimodality, and accuracy. And then once we got there, we actually get our database. And 8 years ago, we started working the Clutera, and from there we acquired over 16 million proteins, and then we annotate all these with different databases, and we ended up with what we call functional dark matter, the things that we can't characterize. And I just want to point out we are working with 2.4 million different proteins, and from which 82% have an unknown function. And we only were able to annotate a very small fraction of those, only about a little more than 100,000. And we have a domain-specific database and a general database. And then the first question is, can we actually understand how architecture influences annotation? And then we take this hierarchy, and we actually developed 16 different protein language models, and with the ones that have the general models that think, okay, more information is better, the accuracy was about like between 0.55 and 65. And when we actually go to the very domain-specific models, we increase this accuracy a lot. And we have one that wins because it has the higher accuracy. So should we abandon all others and study this one, or do we need all the others? So are the models that have the lower performance is still teaching you something about protein annotation? And when we get there, we actually, if we take all the 16 models, we actually are able to find about almost 60,000 novel orthologs based on the protein. So basically, we are able to annotate 10 times more proteins than or almost 7 times more proteins than what we had. And when we actually go to, when I go talk with the biologists and we say, okay, each one of those proteins needs 700 euros to be expressed, which of them we choose? And then they say we go to the 89, and that's why we go for this score of 8,000 that all the models use. And then, but then they ask, okay, but is it possible that a class is not there? And when we ask this question, there are classes of proteins that no model could actually answer. And at the same time, when we think about the best models, are classes that best models were not able to annotate, as well as the other models, and the answer is yes. And then how did it get to what we don't know? So, and this is the part where we think about how to harness the Helmholtz for expertise. In one point, we actually are doing experiments. We go to the lab and we do these things, and we are doing this with Guillemard and Jülich. We actually trying to curate the database even more, and we have a team of, and this is where the 6 different countries come to join, and particularly Guillemard and Jülich are leading these efforts. And then we also go for computational validation of the things, because you can't do everything in the computer, and then the Guillemard and the GFZ are entering that. And beyond that, we also want to understand generalization and specificity of the model, and we are using these laws of Bayes functions to understand this and bring the explainability to the model, and we are doing this together with the Munich and Jülich. And with this, I know it was a very dense presentation, but I would like to open it for questions. Thank you.
Thank you so much, Ulysses. Do we have questions from the audience? Yes.
Yeah, thank you very much, Ulysses, for this talk. I have a question regarding this structure sequence alignment approach, where you include the AlphaFold predicted structure. So how robust or how reliable are the structures if the proteins are longer, or if you have certain structurally conserved elements in the proteins that are not there in all the other orthologs? So how robust is this method against this?
So at the moment, we are using that the probability on how good they are to relate, and we are doing now networks of similarity and 3D modeling. This is the points 3, the one that we do the validation of the function dot marker with Jülich. So we are actually doing these experiments now. We cannot do this for 600,000. We select 7 or 8 different protein types that are interesting. People, plastic degrading enzymes, antimicrobial degrading, and a couple of other things that are easier to test. And in approximately 2 months, I will have the answer for this question, but not today.
Okay, thank you.
Okay. If there are no further questions.
Thank you.
Everyone's digestion. Yes, then we move on. Thank you so much for